wondered if a simple blood draw could bring a long-extinct animal roaring back to life again one day?
Using just a tiny tube of blood to seed a creature that hasn't walked this earth in millennia.
We burst into Jurassic Park applause. Let's unpack what non-invasive cloning really means and what it doesn't promise.
Stick around as we travel from labs to legends, meeting dire wolf pups and debating the ethics with our pack.
We've seen comments calling us robots, so let's address the elephant in the room before diving into the science.
Yes, we sometimes lean on AI voices, but real humans write these lines, and real mammoth genes inspire them.
Non-invasive cloning could revolutionize conservation by making it safer, faster, and more humane, yet challenges remain.
We'll unravel how blood-based cloning works, meet some adorable proxies, and confront the heated ethics swirling around de-extinction.
Ask yourself, just because we can engineer a mammoth, should we? We'll weigh evidence, hype, and caution together.
Drop your questions and predictions in the comments. Our pack's curiosity drives these explorations and shapes future episodes.
Picture a world where a simple vial of blood could secure the future of species we pushed to oblivion. In 2025, Colossal Biosciences announced a breakthrough: cloning animals using blood-derived endothelial progenitor cells instead of painful biopsies.
Rather than darting and sedating rare rhinos or wolves for tissue, scientists could now collect a sample at the zoo.
Ben Lamb called this a critical piece of scaling genetic rescue, and it's not hard to see why. Endothelial progenitor cells normally build blood vessels, but in culture, they multiply quickly and offer plentiful donor nuclei for cloning.
Using somatic cell nuclear transfer, those EPC nuclei are slipped into an egg cell and coaxed to develop into an embryo.
In April 2025, Colossal welcomed 2 litters of cloned red wolf pups, produced entirely from blood samples, boosting genetic diversity.
By eliminating surgery, non-invasive sampling also speeds things up because EPCs grow fast. Time is the enemy for endangered animals.
Imagine field biologists drawing blood from a wild rhino and shipping cells to a lab, skipping sedation and invasive cutting entirely. That shift matters because one stressed animal can be the last of its kind. Minimizing harm is a moral imperative.
Yet, the company isn't stopping at blood draws. It's pouring resources into artificial wombs to free animals from surrogates.
They've developed a microfluidic incubation system with specialized culture medium and hydrogels that let embryos grow longer outside a womb.
For marsupials, the team even built an artificial pouch environment where qual embryos reached halfway through pregnancy without a living mother.
While no mammal has been born ex utero yet, Colossal hopes the first will be a small mammal soon. Importantly, the company states it's not pursuing any human reproduction applications. The focus is strictly wildlife and conservation. They even note that artificial womb technology might one day benefit human medicine indirectly, like neonatal care or improved IVF incubators.
Let's meet the first species in their revival pipeline, the woolly mammoth, the celebrity of de-extinction dreams.
Scientists are splicing dozens of mammoth genes for hair, fat, and small ears into elephant cells, creating cold-resistant hybrids. In 2025, they even birthed woolly mice carrying mammoth-like traits, a proof of concept for bigger beasts.
Colossal predicts its first mammoth elephant calf could arrive by 2028, ideally gestated in an artificial womb to spare elephants.
The company argues these hulking herbivores might restore Arctic grasslands and slow permafrost thaw, though many ecologists remain unconvinced.
Next in the pipeline is the Tasmanian tiger, or Thylacine, a marsupial predator that vanished back in 1936.
Researchers sequenced its genome and created stem cell lines from donor cells, building a genetic template for resurrection.
They've even prototyped an artificial pouch to gestate Thylacine joeys. Progress is ahead of schedule, but challenges remain. Some optimists dream of reintroducing thylacines within 8 to 10 years, yet producing a living joey is still a leap.
Then there's the dodo, a flightless bird whose reproduction requires eggs, making cloning impossible. Instead, scientists edit pigeon germ cells.
The dodo genome has been sequenced, but reaching primordial germ cells inside pigeons is tricky. This project will take longer.
Mauritian officials are already discussing potential habitats for dodo-like birds, though technical hurdles mean they may arrive after 2028.
Finally, April 2025 brought news of dire wolf pups. In truth, they're gray wolves edited at about 20 gene sites.
Non-invasive cloning begins with a simple blood draw, extracting EPCs instead of skin biopsies, which reduces stress and risk. Those EPCs proliferate rapidly in culture. Scientists then transfer their nuclei into enucleated eggs and stimulate them to divide.
Cloning required laparoscopic retrieval of eggs and skin biopsies, plus anesthesia for donors and surrogates. This technique changes that.
For endangered or skittish animals, being able to collect blood in the field could make the difference between extinction and survival.
However, the process still involves somatic cell nuclear transfer and gene editing. It's not as simple. No calf has yet been born outside a womb. The first success will likely be a small mammal, not a mammoth. Replicating the complex physiology of a womb requires controlling nutrients, waste exchange, movement, and immunology. A huge engineering challenge.
Colossal says its first mammoth-like calf could arrive by 2028, but note, this would be a hybrid, not a resurrected mammoth.
Beth Shapiro reminds us, it is impossible to bring back an extinct organism identical to the original. Hybrids are the reality.
Dire wolf pups, Romulus, Remus, and Khaleesi, have over 99% gray wolf DNA and only 20 edits.
Scientists like Peter Mera caution that calling them de-extincted dire wolves is misleading. They are engineered gray wolf hybrids.
Recreating a full extinct genome is extremely challenging because ancient DNA fragments are degraded and incomplete. Guesswork fills gaps.
To call these creatures becomes a philosophical puzzle. Proxies, hybrids, new species, semantics shape how we perceive them.
Cloning's failure rate remains high. Many embryos fail to develop or result in offspring with health problems like premature aging.
Scaling up to large mammals like mammoths magnifies welfare risks, both for the developing fetus and any surrogate mothers used.
Ecological uncertainties abound. Extinct species evolved in different climates and communities, and reintroductions could disrupt or fail in modern ecosystems.
Peter Mara warns that without restoring habitat or removing threats, releasing proxies is not a solution, it's a diversion.
An editorial called Mammoth De-Extinction, Bad Conservation, because funds could better support anti-poaching, habitat protection, or captive breeding.
Philip Seddon suggested some projects are more about fundraising media coverage than solving real extinction, crises, motivations, matter.
Many scientists see value in genetic rescue tools. Non-invasive cloning can bank biodiversity and avoid harming fragile donors.
Cells from old or deceased individuals can be used to clone new offspring, preserving genetic diversity and creating insurance populations. Colossal partners with conservation groups to biobank cells from dozens of endangered species, backing up life in cryogenic archives.
Studying resurrected or proxy animals yields insights into genetics, evolution, and physiology, from fat metabolism to hair growth in extreme cold.
Elephants, a gene more affects us. Elephants have multiple copies of the cancer-suppressing gene P53A. Research on mammoth hybrids could inform human cancer therapies.
Immunity lessons from resurrected dodos or ancient wolves might inform disease research like avian flu or canine genetics.
Some ethicists argue we owe an obligation to bring back species humans drove extinct as a form of atonement.
Seeing proxies roam again could inspire awe and renewed interest in conservation, especially among young scientists and the public. Animal welfare remains a central concern. Cloned animals often suffer miscarriages, deformities, or premature aging, despite improved methods.
Even with artificial wombs, surrogate mothers may still be needed for some species, exposing them to risk until technology matures.
What kind of life will a lone resurrected mammoth or dodo have, without a herd or parents to teach behaviors? A species reintroduced into a changed environment could become invasive or perish. Ecological impact assessments are essential before any release.
Target effects in CRISPR editing and limited genetic diversity could make revived populations susceptible to disease or inbreeding problems.
Ancient pathogens are unlikely, but modern pathogens could hit revived animals harder since they lack immune memory. Biosecurity is critical. Regulatory frameworks must catch up. Cross-border release of CRISPR-modified mammals will require international agreements and transparent risk assessments.
There's a moral hazard. Believing extinction is reversible might reduce urgency to protect habitats now. Technology cannot be a safe.
Opportunity cost matters. The price of resurrecting one mammoth could fund conservation programs for dozens of species currently struggling. Bioethicists caution against a technological imperative. Just because we can, doesn't mean we should. Prudent vigilance is vital.
Indigenous peoples, like some Maori voices, have expressed concerns about resurrecting ancestral animals without consultation. Cultural respect is crucial.
Summing up, the science is moving fast, but it's filled with uncertainties. Progress should be paired with humility and oversight. Remember, non-invasive cloning gave us cloned red wolves from blood and dire wolf hybrids with 20 edits in 2025 alone.
Recap: Mammoth elephant calf projected around 2028. Thylacine 8 to 10 years. Dodo further out due to germ cell complexity.
These projects integrate ancient DNA analysis, CRISPR editing, cloning, and artificial womb research. It's a systems-level enterprise.
We hope you're following along. Pause and review the sources in our notes panel for deeper dives and timestamped references.
Question or source of your own?
Share it.
We'll highlight your insights.
And pivot to our debate next.
Wolf, one of America's most endangered predators, with fewer than 24 in the wild. Colossal used non-invasive blood draws to create cell lines from 3 individual wolves, then cloned litters to increase diversity.
Those pups are genetically identical to their donors, providing a safety net if disease or disasters strike the dwindling population.
Because the cells came from simple blood draws, no wolves were anesthetized or harmed. This matters for skittish, rare animals. Early reports suggest the cloned pups are healthy, but long-term monitoring will reveal whether cloned predators thrive and reproduce naturally. Our second case is the dire wolf pups. They're gray wolves edited to express a handful of ancient traits. Scientists identified about 20 genes related to size, coat color, and other features from fossil DNA, then used CRISPR to edit embryos.
The edited embryos were implanted into domestic wolf or dog surrogates, resulting in pups with larger size and paler coats.
Despite headlines, the pups retain over 99% gray wolf DNA. This underscores the complexity of resurrecting a species fully. Still, seeing their snowy coats and slightly bulkier frames reminds us how powerful gene editing is, even when results are partial. Our third case goes bigger. The Woolly Mammoth Project aims to splice dozens of cold adaptation genes into Asian elephant cells. These genes code for long hair, subcutaneous fat, and small ears, traits that help mammoths survive frigid Ice Age climate. Is to produce a cold-resistant elephant-mammoth hybrid calf in 3 to 4 years, ideally using an artificial womb. Now, let's examine the Thylacine, known as the Tasmanian tiger. This striped marsupial vanished less than a century ago. Colossal sequenced the Thylacine genome and compared it to the Dunart, a related marsupial, creating stem cells for editing. They've designed an artificial pouch environment to gestate joeys, mimicking a marsupial's unique developmental journey in the wild.
Optimists speculate thylacines could return to Tasmania in 8 to 10 years, yet real birth and rewilding hurdles loom.
If revived, they'll need habitat corridors, prey populations, and protection from competitors. Reintroduction isn't just a lab problem. Australia's indigenous Palawa people would also deserve a say. Cultural considerations will shape any thylacine reintroduction plan. Moving on to birds, cloning doesn't work for eggs, so Colossal plans to edit primordial germ cells in the Nicobar pigeon. Germ cells, the precursors to sperm and eggs, are harvested from embryos and cultured before being edited with Dodo DNA.
The edited cells are then transplanted back into pigeon embryos, producing birds whose gametes carry Dodo-like genetic instructions.
Once those pigeons mature, their eggs and sperm could yield chicks with Dodo traits. However, the technique is in early days. Mauritius is preparing habitat plans for potential Dodo return, working with conservationists to ensure ecosystems can support them. Beyond the flagship projects, Colossal has floated reviving the New Zealand giant moa and even the Stellar's sea cow. The moa has no close living relative, so creating one would likely require a fully artificial womb and unprecedented gene synthesis.
Critics argue such ventures have little to do with solving extinction crises and more with headlines. They caution focusing on present species.
Still, Discussing them illustrates how far technology might stretch and reminds us to ask where ethical and practical lines should be drawn. Species without close relatives emphasize the importance of artificial wombs. This research is crucial, even if de-extinction never happens. Non-invasive cloning isn't just for extinct icons. It's being used to save ferrets, rhinos, and other endangered animals. By collecting blood from the last surviving rhinos, conservationists can clone genetically diverse calves and buy time for habitat recovery.
Cell banking programs act like Noah's Arks for genetics, preserving tissue from dozens of species for future cloning or editing.
Combined with AI-driven wildlife monitoring, non-invasive cloning could become part of a broader conservation toolkit that includes drones and bioacoustics. These examples show the science isn't just theoretical, it's happening now, and we can participate by staying informed and engaged. Across these cases, non-invasive sampling and gene editing are keys, but each species presents unique technical, ecological, and cultural hurdles. Red wolves show immediate conservation benefits. Dire wolves reveal limits. Mammoths push engineering. Thylacines and dodos highlight marsupial and avian complexities.
The moa and sea cow remind us that some de-extinction dreams may be unrealistic until artificial wombs and gene synthesis advance.
Through each story, the interplay between wonder and caution surfaces again. Our next section celebrates that community dialogue. But first, let's pause for a moment to acknowledge the voices that shape our channel. Your voices. Our community often asks tough questions, like @FractAlchemist, who worries revived species could act like invasive species in fragile habitats.
It's a valid concern. We responded by exploring how ecological impact assessments and containment plans would be essential for any reintroduction.
Then, @Kindred Creatures dove into the nitty-gritty of pigeon cell editing, asking about timelines and first hatch expectations for dodo chicks.
We shared a roadmap. Isolate primordial germ cells, edit them, transplant them back, then wait for the next generation to hatch.
Your curiosity set expectations. We asked the pack what success looks like. One chick, a stable flock, or a thriving wild population.
Viewer @PietStridon reminded us that beavers are the original ecosystem engineers. We agreed and promised more beaver content soon.
Meanwhile, at 3 Days of Darkness, C2Y criticized the $15 million budget for the MOA project and demanded accountability, sparking an important discussion.
We clarified the funding was private and covers genome assembly, CRISPR work, surrogates, and animal care under Maori leadership oversight.
Some of you took our Cun of the Week seriously. @Musketeer Chris quipped, "You can't spell determined without ermine," and we howled.
We replied with a de-erminid pun and explained how thermal cameras detect heat rather than color in stoat hunts.
Your feedback about synthetic voices also hit home. @FrogBear02 vowed to stop watching until we sounded authentic again.
In response, we promised behind-the-scenes segments showing our tiny human team and how AI helps amplify underreported conservation stories.
Viewer @RichardG1PQ sent photos of 5-toed tracks claiming live thylacines. We asked for clear scale, track lines, and terrain notes.
We're compiling credible leads for museum teams, but remind everyone that dew claws, double registrations, and soft ground can create confusing prints.
Many of you also loved our micro shorts, like the Amber Fossils episode. Enumerated titles and curiosity hooks boosted click-through rates.
We're learning from analytics. When average view duration dips, we insert debates. When CTR falls, we test new thumbnails and polls.
Our poll comparing wolf, coyote, and husky translations showed coyotes as the crowd favorite. Expect a comparison short soon.
We also plan to feature fossa translations, thanks to @Musketeer Chris. 4 fossa pups were born recently, inspiring hope.
Join our citizen science map by uploading photos of tracks, nests, or sightings. We'll collect data and share patterns with researchers.
Remember, include location and timestamps when you submit field notes. Good data means better science and fewer false alarms.
Your stories of helping rescue injured wildlife or planting trees and mangroves leave us hopeful and grateful. Please keep sharing.
Our PAC's passion fuels this channel. Like, comment, and subscribe if you want more science with empathy and transparency.
Now, let's look ahead to the future of de-extinction technology and how it could reshape conservation and medicine.
Artificial womb research will continue refining nutrient delivery, waste removal, and oxygen exchange, aiming to support embryos through full gestation. Scaling these systems to support an elephant-sized fetus is a major engineering challenge, but successes with smaller mammals pave the way. Once reliable, artificial wombs could carry multiple embryos simultaneously, accelerating population recovery and significantly reducing risk to surrogates. Some hope mammoth-like herds could help maintain permafrost by trampling snow and fertilizing grasses. But ecological benefits remain speculative. Non-invasive cloning techniques might become routine for conservationists, empowering them to boost numbers of rhinos, ferrets, or other species from banked cells. Gene editing could help living species adapt to climate change by adding heat tolerance or disease resistance, raising new ethical questions. International guidelines will be needed to govern releases of CRISPR-created animals across borders and ecosystems, ensuring accountability. Advancements in artificial wombs and gene editing may spin off into human medicine, better IVF incubators, organ growth, or cancer therapies. We'll also see AI integrating with these fields, from monitoring surrogate-free gestation to predicting off-target effects using machine learning. The coming decade will test not just our technology, but our moral compass. We must decide what kinds of creatures to create. I'm cautiously optimistic. With robust oversight, these tools could complement, not replace, habitat restoration, anti-poaching, and community-led conservation. Research on marsupial pouches might even improve neonatal care devices for premature human babies. Cross-species engineering goes both ways. Indigenous consultation will be critical, ensuring communities whose ancestors lived with these species have a voice in revival decisions. We'll continue covering breakthroughs and controversy. Send us questions about artificial wombs, gene editing, or regulation for future shows. Up next, let's condense all this into actionable takeaways and a hopeful close. We value your time and your passion. Remember, non-invasive cloning uses blood-derived endothelial progenitor cells to clone animals, avoiding biopsies and reducing stress for endangered donors.
De-extinction creates proxies, not perfect copies. Dire wolves are gray wolves with edits, and mammoth elephants will be hybrids.
Ethical guardrails matter. Consider animal welfare, ecological impact, moral hazard, and opportunity cost carefully before supporting resurrection projects.
At the same time, these tools could save living species and inspire awe. Support local conservation, citizen science, and community engagement.
Share your hopes and concerns in the comments. Like and subscribe, and stay tuned for Colorado wolves and quantum sensors.